What we have seen that when a pentose sugar, this is the pentose sugar, when a pentose sugar attaches to one nitrogen base at one position, then it is called nucleoside. And when it's C5 attaches to the phosphate group, the CH2 we can write here, CH2OH was there. CH2OH में से ये पूरा OH और एक वाटर मॉलिक्यूल बनने के लिए चाहिए 1H. 1H is taken from the phosphate group. Then one bond is formed between the phosphate group and this sugar. So this CH2 is remaining, that's why we have written this CH2 and then this phosphate. The phosphate at C5 position and the nitrogen at C1 position. So this is a nucleotide. Fine. Now we will see how from a nucleotide the polymer is formed. First we will see the one strand of DNA. Very simple actually. What we will do? बहुत सारे जुड़ते जाएंगे और एक स्ट्रैंड बनते जाएगा. तो चलो बनाते हैं ना एक स्ट्रैंड. 3 बना लेते हैं शुगर. ये एक, यहाँ पे नाइट्रोजन बेस कौन सा लिखे? चलो सी ले लेते हैं, साइटोसिन. हियर राइट, हियर वी हैव रिटन CH2 एंड P. दूसरा ले लीजिए. दिस इज अनदर वन having, say here it is the T1 and here again CH2 and P. Now third molecule फिर से बनाया ऐसे और यहाँ जोड़ देते हैं A एडेनिन और यहाँ पे CH2 and P. The only thing which we have to do, see, this is one nucleotide, this is another nucleotide. Let me draw the second nucleotide in a different color. Okay. So this is the sugar. Here there is, say, G is there. Here CH2, OH is no more there, and the phosphate group. Now this, what is this position? This one. This is the nucleotide. This is its 1 carbon 1 position. This is 2. This is 3. 3 वाला OH उसने free रखा हुआ था एक दूसरे के साथ जुड़ने के लिए. So now this phosphate, it will join to the third position carbon of जो nucleotide उसके ऊपर है, उसके third carbon से ये जुड़ गया. This bond between the nucleotide of phosphate and carbon was called ester bond. अब ये ही phosphate दूसरे nucleotide के C3 पोजीशन से जुड़ा है, तो ये दोनों बॉन्ड मिला के कहते हैं, yes, it is called phosphodiester bond. So this is called phosphodiester bond. जैसे यहाँ P को जोड़ा था, सेम अब ये P को ये इसके C3 के साथ जोड़ देंगे. तो this one and this one, these two bonds, they are again the phosphodiester bond. बन के तैयार. ये strand बन के तैयार हो गया. ऐसे लाखों जुड़ते जाएंगे and one strand of DNA will be formed. अब आप ये देखिए, ये जो न्यूक्लियोटाइड्स है, यहाँ पहला वाला जो स्टार्टिंग है उसका ये कौन सा कार्बन का ये OH एकदम फ्री है. This is 1, 2, 3. Here this position is free. This is called 3' end. This is called 3' end. और एकदम लास्ट में यहाँ देखिए, let me remove this one. Okay. So यहाँ देखिए, ये पांचवें साइड में ये फॉस्फेट. अब फर्दर और कोई न्यूक्लियोटाइड वहाँ तो है नहीं, ये आखिरी वाला है. तो ये P यहाँ पे फ्री है. दिस इज द 5 प्राइम एंड. दिस इज ये CH2OH पांचवा कार्बन था, ये 5वां साइट है और ये तीसरा है. Now the next strand of DNA. We know, Watson and Crick knew that C always joins with G. C always pairs with G. They found out that it joins by 3 hydrogen bonds. G always pairs with C. A always pairs with T with 2 hydrogen bonds. So here we have drawn 3, here we have drawn again 3, and here we have drawn 2 bonds between A and T. So this GCT, it is attached to the nucleotide of the another strand. दूसरा strand बना दे. बनाते हैं हम DNA का दूसरा strand. Already I have written here the nitrogen bases which are present in the next strand. So you know that this nitrogen base is attached to the C1 position. One thing, same nucleotides you have to draw here, लेकिन एक चीज याद रखनी है that these 2 strands are anti-parallel. मतलब इसका अगर 5 यहाँ है तो इसका 5 इस साइड में होगा. So इसका ऑक्सीजन इधर है तो आपको ऑक्सीजन का मूव टर्न करना है. So you have to draw it this way. So this is G, that is C1 position. यहाँ से आपने ये लिया 1 2 3 and then 4 and then it comes to 5. So the phosphorus is of interest. We will draw it here. Now this next nucleotide again. This is the cytosine. Now this is carbon 1, 2, 3, 4 and this is 5. Done. Now here again this is the complete nucleotide, the phosphorus we have drawn. Now the next one, this is the thymine. Here it will be attached to the C1. So here the C1, 2, 3, 4, 1, 2, 3, 4 and this is the 5th with oxygen. Here the last phosphorus which is not attached to any other if you consider it as the last one. Okay, now we simply, this is the 3 dash end, 1, 2, 3. So we write here 3 prime. This is 5 prime, this is 3 prime. This is called, the 2 strands are anti-parallel. They are parallel but their polarity is in opposite direction, so they are anti-parallel. This is the phosphate which is attached to the C5. This is its 5 end. This is called 5 prime. See, both the strands of DNA are ready now. Okay, so you know that DNA is not this, does not have, the 2 strands do not run straight. They run as a helical structure. When they run as helical structure, then they are forming this structure. This much, this width of the DNA, ये जो DNA का width है, this width is 2 nanometer. This width is 2 nanometer. This width is 2 nanometer and the one turn of DNA, DNA के एक टर्न में, मतलब ये यहाँ से एक स्ट्रैंड शुरू हुआ, ये ऊपर गया, ये नीचे आया. This one turn is 3.4 nanometer. और इस एक टर्न में होते हैं 10 पेयर ऑफ नाइट्रोजन बेसेस, यहाँ पे 10 पेयर ऑफ नाइट्रोजन बेस है, पूरा 10 मिलाने के बाद फिर जो डिस्टेंस आता है दैट इज 3.4 नैनोमीटर, इसीलिए ये दोनों के बीच का डिस्टेंस होगा 3.4 डिवाइडेड बाई 10 इज इक्वल टू 0.34 nanometer. That is all about DNA structure. We will summarize now what we have studied till now, that we will summarize. Now you know all the complete structure, so now it will be easy for you to understand. Okay, now let us simply summarize what we have studied. Ready? So you know that DNA, it consists of 2 strands. So these 2 strands, though they are parallel to each other, they are not straight. They are helically wounded around each other. If this is 5 and this is 3, this is 5 and 3. Okay, 2 strands helically wounded and antiparallel, having opposite polarity. This 5 this side, another's 5 this side. Good. Now this width is 2.0 nanometer. This width is 2.0 nanometer and the width of DNA is 2 nanometer and 1 turn. What is 1 complete turn? Take any one strand. If you put finger here, okay, now it is moving up, it comes here. This is not 1 turn, this is half turn. It moves down and here it completes 1 turn. This much, this is 3.4 nanometer. This is 3.4 nanometer and in this 1 turn there are 10 pairs of nitrogen bases in it. So let us draw 10. 1, 2, 3, 4, 5, 6, 7, 8, 9 and 10. So there are 10 pairs of nitrogen bases. So naturally distance between these two will be 3.4 divided by 10, that is 0.34 nanometer. Now in this strand, where is nitrogen, where is sugar and where is phosphate? Let us make it a straight one, a simple to understand one. In these 2 strands, you know that there are 4 nitrogen bases, but if you are going to write this common mistake when the students do, so first let us do the mistake so you never repeat it. If we draw here the nitrogen bases, see here, so G, A and T and A, G and C, this will be a wrong one. This diagram is a wrong one because these 2 strands, which are called the backbone of DNA, it is made up of only alternate sugar and phosphate molecules. It is made up of alternate sugar and phosphate molecules. And where is nitrogen then? Let us see. So this is sugar, phosphate, alternately they are there. To understand this, you can here I am drawing the two nucleotides joining together which are forming the DNA strand. इसे देखोगे तो ये समझ में आएगा. जैसे ये एक न्यूक्लियोटाइड है, ये शुगर के 5वें पोजीशन में फॉस्फेट है, 1 नंबर पे नाइट्रोजन बेस है. This is phosphate, this is nitrogen. Similarly here also, C1 joined to nitrogen, C5 joined to phosphate. So here this sugar and phosphate is a part of nucleotide. इसमें नाइट्रोजन बेस जुड़ेगा तो न्यूक्लियोटाइड बन जाना है. But nowhere you see that this phosphate is joined to its own sugar, and it will join to the C3 position of the next sugar. So phosphate group by phosphodiester bond is attached to two sugars. Nowhere it is joined to the nitrogen base. These two are at a distance. ये यहाँ है और ये यहाँ है. So you draw sugar phosphate means you are drawing sugar phosphate like that. From the sugar, if one is having C, another will have G, and in between there are 3 hydrogen bonds. Leave aside phosphate, come to the next sugar. If this is G, this is C, 3 hydrogen bonds. If this is A, this one is T and 2 hydrogen bonds. So this is the structure of DNA, and looking at this model which they had given, Watson and Crick, when they looked at it, they could hypothesize the next thing. एक्सपेरिमेंटली प्रूव नहीं किया अभी तक, अभी तो बस स्ट्रक्चर का मॉडल दिया है ना, मॉडल दिया है. बट लुकिंग एट इट उन्हें ये समझ में आया कि हाउ द डीएनए माइट बी रेप्लिकेटिंग मीन्स व्हाट. एक पेरेंट है, अब रिप्रोडक्शन में जब वो ऑफस्प्रिंग बनाएगा, सो फॉर द ऑफस्प्रिंग इट नीड्स अ सेट ऑफ डीएनए, तो वो एक कंप्लीट अपनी कॉपी बनाता है, एक रेप्लिका बनाता है. दिस इज कॉल्ड रेप्लिकेशन. या एक सेल है विच इज गोइंग टू डिवाइड टू फॉर्म मेनी सेल्स. एट दैट टाइम ऑल्सो टू गिव द सेल्स इट फॉर्म्स एग्जैक्ट रेप्लिका ऑफ इट्स डीएनए एंड गिव्स इट. सो वो एग्जैक्ट रेप्लिका में भी कुछ गड़बड़े होती है, वो दैट वी विल सी आफ्टरवर्ड्स, बट इट प्रिपेयर्स अ रेप्लिका ऑफ इटसेल्फ. And how that replica is formed, that is the next question. They looked at it and they said the DNA must be replicating by semi-conservative type of replication. What is semi-conservative type of replication? Can you also think of yourself as Watson and Crick? Look here, and can you hypothesize what could be semi-conservative type of replication? This we are going to see how Michelson and Stahel they proved in 1958 that yes, it really is the semi-conservative type of replication by which DNA replicates. See you in the next video for the semi-conservative replication of DNA.